All right, so now let's discuss the motor units. I just try to trace my arm a little bit, but that's a little bit exaggeration. But this is going to be really interesting in this conversation about what the motor unit is. And so the very first thing you need to really know is that definition of a motor unit. And remember that the motor unit is the single alpha motor unit and all of the fibers that it innervates. All right, remember that definition is going to be very important. And that motor unit is basically it's the functional unit of the whole skeletal muscle system. Now contrast that to the sarcomere that we talked about before. The sarcomere was the smallest contractile unit of the muscle cell, right? But the alpha motor unit, that is, as I said, that's the basic functional unit of the whole skeletal muscle as an organ, okay? Now, also what I drew here is, this is your whole entire muscle, but there's a number of alpha motor neurons that go to those different muscles. In fact, the human body has like over 420 different neurons, and essentially billions, like 1.4 billion muscle fibers. So it's an insane amount. And this helps you also understand this is like there's a nerve fiber ratio. So again, this is just one, like this is your bicep muscle, but you have different motor units and in your entire muscle, right? It's not just one, it's not the entire muscle. And there's these different ratios, this fiber to nerve ratio basically. And this is the innervation ratio. So you can have a low fiber to nerve ratio, so say like 2:1 or 10:1, and that's for like very precise movements that you're going to do, like moving your finger, or you're playing the piano, or even like your eyes, right? All of those have a very low fiber to nerve ratio. But your other things have a very high fiber to nerve ratio, where there could be like 500 to 1 or 2,000 to 1, and that's for your gross movements like jumping or kicking a ball or something like that, right? So that's the biggest difference with that. Now, when it comes to the different motor units, they also have different sizes, and the size of the motor unit is related to the innervation of that functional role of the motor unit itself. So if you have like a smaller motor unit, those ones are connected to fewer fibers, and therefore they're motor units with a low innervation ratios, and they're involved with the precise movements like playing the piano that we talked about. And then the larger motor units, those are connected to more fibers, and therefore they have more a higher innervation ratio, and that's going to be your gross movement, right? That's your jumping. Now the motor units is the way that this can be distinguished, like when we look like on our microscope, is a lot of it is the soma of the motor unit, or the cell body basically, and that resides in the spinal cord itself. Now the small motor units, they have a lower recruitment threshold due to a small soma. Remember we drew the soma, and they have the EPSP and IPSP and all of this going on. So have a smaller soma, then it has a lower recruitment threshold due to the smaller soma. But the larger motor units, well, that has a higher recruitment threshold due to its larger soma. And then the action potential from the interneuron will activate the smaller motor neuron before it activates the larger one. Okay. Now there's different types of motor units. Then essentially you're going to have your fast twitch, and then your intermediate twitch, and your slow twitch. Now these are your motor neurons, okay? So we could just write this. Okay, so like we have our FF, this would be our fast fatigable, and then we could write our FR, fatigue resistant, and then we have our slow, like slow twitch basically. And of course, is implied, the fast fatigable, yeah, they're going to be super fast. They're going to produce a lot of force very quickly. They relax fast. They fire rapidly, and so on. And then your fast resistant or fatigue resistant. Those are your fast twitch muscle fibers, but they're sort of fatigue resistant because they have more aerobic activity to them, and so they're able to produce a lot more force than, say, the slow twitch ones that are very fatigue resistant, but not as much as the fast fatigue ones. Okay, so there's different. Again, these are motor units, and it's correlated tightly to your muscle fibers. But there's a distinction between the motor units and the fibers that we're talking about. All right. Now, each one has a different force characteristic to them. So if we just drew like a graph right here, and if we did think about the fast fatigue ones, okay, so here you get if we put force right here, it's going to be measured in newtons, and then we put our time here, and for the fast fatigue, we can get something like, boom, it shoots up lots of force and also very fast relaxation, and it goes back down. Now, the fatigue resistant, it's going to shoot up very fast, right? Go very high, and it's going to come back down, and there it is, okay? Now your slow twitch, that's going to be like, oh yeah, I'm doing it, I'm coming, right? And it doesn't quite put as high, and then it's going to come back down. There's better diagrams in the slides that you have, but this gives you an idea of the difference between each one, and understanding the myosin heads and the parvalbulin, for example, that can quickly get rid of that calcium. That's going to be important. And this is when essentially all your individual twitches, they kind of fuse together because you have rapid succession of the alpha motor neurons firing, and you don't allow your muscles to relax all the way. And so they kind of fuse all together. And when those twitches overlap, you get tetany, you get tetanus. And that's what that is. And so we could actually draw this. Maybe we'll just do it right here, what that might look like. Okay, so here we have our, I guess on the y-axis we can do our tension of strength. Okay, so we'll just put, okay, our tension over here, and then I'll put our time right here. And first we get a muscle twitch. Okay, so we get a muscle twitch, it's come down, and there it is. That's just a muscle twitch. Okay, the contraction and the relaxation. And now we can start to fuse some of these together. So maybe we get two consecutive twitches, two consecutive stimuli together, and now we're going to fuse some of this together. So in this case, maybe it'll. Well, let's see. Yeah, we'll follow the same. So it comes up right like this, and then it starts to come back down. But then another active potential comes, and so now this one starts to fuse together, and then it's going to kind of maybe stay right here. So now it's not reaching max tension, but they're kind of two of these are fused together. So we get this consecutive type stimuli, and it's an unfused one. Now, if we get like a whole bunch of that stimuli that's coming in over and over again, now we can really take this and put max tension, and then it just stays right here. So like that's just like totally contracted, just, and you have max tension because, again, you have your alpha motor neuron. That neuron is depolarizing, it's repolarizing, acetylcholine is getting released. The acetylcholinesterase is getting rid of that acetylcholine, but all that happens, that calcium gets pumped in. but you're getting another action potential in the acetylcholine release before you're able to get all that calcium to be uptaken into the sarcoplasmic reticulum, and it's using a lot of your ATP, so all that calcium is just staying bound to troponin C, therefore you're getting this cross-bridge cycling, and you just keep on going on and on and on. And so you end up getting this long tension that's occurring, and it'll stay that way until what? Until you literally start running out of ATP. Actually, it's not entirely true, but as an idea, you start running out of ATP. It's actually not what's happening. There's a lot of ATP still left, but remember those circuit pumps are using a lot of ATP, but what's going to happen is fatigue is going to start setting in. We'll talk about what the causes are fatigue later, but it's not really ATP depletion. But then this is going to start decreasing, right? And that's going to be the fatigue. So that's the tension that can happen. And now we can understand this is a similar concept we talked about with the EPSP and IPSP with a summation. In this case, we're going to call it gradation. And this is basically the series of successive stages of the strength and that's required for the coordinated contractions that's going to happen, right? So like how is the strength of muscle contraction graded, basically, right? So you can have smooth movement. You're going to pick like this pen, I'm going to pick that up. You've got to be able to grade all of these, and this is how it's done. One of them is through spatial summation, basically, and that's the multiple motor unit summation, because again, we have multiple motor units, and we can activate the smallest ones activate first. We can activate a little bit more, and all those muscle fibers are going to be activated. That's one thing I want to make sure you remember too, is that all or none principle, right? When this alpha motor neuron gets activated, all of the muscle fibers that are within that motor unit are activated, right? Not just a couple fibers, all of them are activated. That's the all or none principle that you want to understand why it is that we can do this spatial summation. We can activate multiple motor neurons, and we can activate more of those motor units and get more of those fibers involved in the contraction process. The other one is simply the wave summation, and that's the frequency summation. That's kind of analogous to our temporal summation with the EPSP and IPSPs, but we talk about them differently. So in this case, we're just talking about it as wave summation or frequency summation. And that's simply when you have the alpha motor neuron is firing on that muscle cell saying, contract, and we're starting to get more and more tension. Maybe we get some more of the tension fusing together, and that increases the tension over time before it allows the muscle to fully relax, okay? Now, as we've talked about earlier, when it comes to the getting muscles contract, right? You have to follow the order of operations, right? Because first you have to get the depolarization of the alpha motor neuron, and then you've got to get the calcium to be released. And after the calcium is released, that's finally when you get the muscle to contract. So you could draw that also. Like we'll just draw something right here really quick, right? If you think about what's happening, you get if this is time right here, okay, you can think about you get your action potential that happens like right here. It's not drawn as an action potential, but you get your action potential, and then it's like right after that, right? You're going to get your calcium is going to happen, right, and go back down. But it's right after your calcium, that's when you start getting your muscle contraction that's going to happen. So I'm just trying to put in order of sequence of what's happening, right? Obviously, the calcium has to precede force generation because you've got to get that calcium to get out of the SR and bind to the troponin, and then you can get the tropomyosin off of actin, and you can start doing the cross-bridge cycling, all right? Now, when it comes to the muscle fibers, there's different ways to classify the muscle fibers. Here, we just classified the motor units, okay, the actual motor units themselves. But the muscle fiber types can be classified into, basically type 1, type 2, and type IIx. And maybe I'll just erase this here. And because I want to talk about just some of the characteristics of these muscle fibers. Now, we're mainly focusing in humans, okay? So we'll go type 1, type 2. Let's be specific. So type No, I'll do type 2A first. Type 2A and type 2X. All right. Now, each one of these have different characteristics. Our type 1 muscle fibers, that's your, if we look at it, that's your red muscle fibers. It's red because they're very oxidative. Well, oxidative requires what? The mitochondria. So if you think about your red meat, like let's say like a chicken leg, for example, right? The legs of the chicken, well, what is that chicken doing? It's walking around all the time on the legs, and it doesn't require, really fast movements. And so it's going to be really rich in mitochondria. It's going to make that meat darker, and therefore it's going to be more like red or darker looking because it has a lot more mitochondria. And that's going to be mainly type 1 muscle fibers. Versus, say, the chicken breast, well, what color is that? It's white. Why is it white? Because those chickens need to fly, and they don't really fly, although we won't talk about that. If the muscle fibers don't have a lot of mitochondria, they're moving very, very fast, and that's going to make it so they don't have a lot of mitochondria in them, therefore it's going to be white. It's going to be more white. That's going to be more of your type 2 muscle fibers, your fast twitch muscle fibers. So just get an idea, right? So again, your red one's going to have, it's more slow, it's oxidative, it's slow twitch, it's that's your type 1 muscle fibers versus your white, it's your fast twitch muscle fibers, and that's your type 2. And you can separate the type 2 muscle fibers into basically 2A and 2X. There's also these type 2B muscle fibers, but that's only in animals. We don't have that in humans, so don't get confused. A lot of confusion out there. Just focus on keeping it pretty simple right here with these methods. And each one of them have different myosin isoforms. And the actin-myosin ATPases are going to be able to basically consume ATP a lot faster or at different rates depending on the muscle fiber type themselves. So like this is basically the KCAT, the turnover in enzyme kinetics. We would talk about the KCAT. And the fast, like the type 1 muscle fibers, I mean, they're going to be a lot slower. Like they're going to do like your ATP, like 300 times a second or whatever, versus like your type 2 muscle fibers could be twice that, right? 600 times per second, that turnover. Now, it's interesting that your slow twitch muscle fibers, they use less ATP because they're more efficient, but they also produce less force, okay? And the speed that you're doing this is really fast because you're basically, the contraction time of the muscle fibers for the fast fibers is like really fast, like 40 to 90 milliseconds, while the contraction time for your slow fibers is like twice that, right? 90 to 140 milliseconds or so. So it's a pretty big difference. Now, you could imagine if you drew kind of a graph of what this would look like here. Again, just let's put our tension, okay? So our tension right here, and I put our time right here, and we can look at our fast twitch muscle fibers, and we would have fast twitch, a lot of force, and it would come down pretty quick, okay? And then we can do our slow twitch muscle fibers, and what you can predict what that's going to look like. Well, it's going to come up, maybe won't go as high, and it's going to come back down, all right? Again, it's going to relax faster because you have parvalbulin, for example. It can grab onto that quicker. You have more of the circuit pumps. You have it's just a higher density of all these pumps to make these move faster. Now, we can look at the actual muscle fibers because remember, there's a distinction between the muscle fibers and the the motor units that control those muscle fibers, okay? Now, when we look at the muscle fibers, we do that by a muscle biopsy, which is basically it's this little, you can see, you can look it up, but there's these tool that you put into the muscle and basically take out a chunk of the muscle fibers. Then you can go into the lab and you can look at the muscle fibers themselves and you can determine which muscle fibers it is. And when you are going to determine the different muscle fibers, there's different ways that has been done, such as gel electrophoresis. And this one has there's mixed homogenates when you do this. You have a lot of things with gel electrophoresis, and then there's also like staining of the ATPase activity. those are pH dependent, and it's done for various like muscle cross sections. And then there's like even mass spectrometry, which is another interesting one. But the main one is immunohistochemistry, and that's kind of considered the gold standard right now. And this just uses antibodies to detect the myosin isoform. All right. Now, when we think about the different proportions of the muscle fibers themselves, well, on average, the average person is going to have about, I'm going to write this here, has like 50%. So your muscle fibers of the average person you're about 50% type 1. And then your type 2 muscle fibers is going to be around 40%. And your type 2x muscle fibers is going to be the remainder, so around 10%. And it's going to be like, it's going to vary, plus or minus 5-15% or so, but this is just the average person. Now, the elite level athletes, and there's the whole nurture versus nature, they can vary quite a bit. So you have like your elite, like let's say a sprinter, for example. Well, they can have a lot higher type 2 muscle fibers, where like closer to 60, 70, 80%. And then the opposite is case for like the marathon runners, right? And then also I want to say it depends on which muscle we're looking at directly. Like some like the gastrocnemius muscle is going to be more one way just from everyday activity versus say the bicep or the rectus femoris or something. So it actually can depend, but on the average person in general, it's going to be at a certain way. Now, we can continue thinking about some of the characteristics of these muscle fibers, right? So remember, just a couple things to think about. And this should all make sense to you with the different properties of each of the muscle fibers, right? So like if we think about the size is going to be something about these. You can think about, say, the SR, the sarcoplasmic reticulum inside of him. You can think about the mitochondrial density. Okay, so mitochondrial density. You have maybe like blood supply. Okay, so you can put capillaries. Your capillaries. What are the things that are in there? Your myosin ATPase activity, oxidative enzymes. Okay, so I can put myosin ATPase. I guess oxidative enzymes would go like with the mitochondria, but we can put like a full point with all of these, as well as fatigue. It's anaerobic capacity and so on, and then we can go through and think, okay, where would you expect this one to be? So like fiber size, for example, well, this is going to be smaller, right? So we can just you think, okay, this will be more small. This one's going to be more intermediate or larger. Well, it's a type 2x, so yes, it'll be large. This one will also be large, and then you can do the same, like hook up with the sarcoplasmic reticulum, right? Well, type 1, you don't need a whole bunch, so you're going to have less of that compared to over here, right? You're going to have a lot more in these ones. You can have like the mitochondrial density. Well, this is going to be super high, right? Maybe I can just draw arrows. This will be super high. This one, yeah, you're going to have them. not like super high compared to this one, but it's still pretty high, because this is kind of analogous to your fast fatigue resistant, but still not as much as this one. But then your type 2x, it's going to be low. You're not going to have very much in that at all. So maybe arrows aren't the best way. Capillaries, you can follow the same idea, right? This is going to be a lot more. You're going to have pretty good, but not as great, and then it's going to be lower with the type 2x muscle fibers. Glycolytic enzyme activity, of course, you're going to be much higher with the type 2 muscle fibers. Your oxidative enzyme activity is going to be much higher with the type 1 muscle fibers. Okay, so just really think about the muscle fiber types and understanding your bioenergetics of what would be what, right? Now, there's some other interesting facts about when it comes to these different fibers, right? Because, again, as I mentioned earlier, your endurance and power athletes, they have a unique fiber composition. In humans, when you're training, you can, first, when it comes to the fiber type itself, it's really the alpha motor neuron that's going to dictate the fiber type. Okay, the alpha motor neuron is what is going to dictate. So you have, like, as I said here, your fast, fatigue resistant. That's your alpha motor neuron. And if that's controlling your muscle fibers, well, then guess what? That's the type of muscle fiber that it's going to develop those characteristics, because that's the signal that's being put onto that. However, what happens with training? If you're like an endurance marathon runner and you're just doing these long, slow miles, well, slow isn't like, a 5-minute mile for 26 miles, not, these super fast sprints. But if you're doing that, you're going to develop a lot more of your high mitochondrial capillary density. And so you're going to have more of this fiber type characteristics that are closer to your type 1 muscle fiber. So you could have an alpha motor neuron that is more like your fast type, but you've changed that muscle fiber characteristics more to be more like your slow twitch muscle fiber. So they can kind of go back, like from type 2 to type 1. It's a little bit interesting. It doesn't seem so much that you can just take your type 1 and go more to your type 2. That one is still kind of debatable. That's not changing the alpha motor neuron. That's just changing the characteristics of the muscle fibers themselves. And so you can, but we've seen this like with various cross-intervention studies where you're doing like chronic stimulations or loading with these studies, and you can see those changes from type 2 to type 1 muscle fibers. And then when you're doing your muscle training, like this guy over here, when those muscle fibers are growing, the muscle is getting bigger, that is primarily, or not exclusively unless you're on anabolics, hypertrophy, not hyperplasia. And your type 1 muscle fibers, another interesting part about this is because they're more, well, maybe perhaps because they're recruited more often, they seem to be maybe more susceptible to injury. But there could be other factors as well with that. And then your type 2 muscle fibers is interesting because you tend to lose these ones more with age. As you get older, you just start to lose more and more of your type 2 muscle fibers, which is maybe a reason why it's good to strength train, you do your sprints, do these power type exercises to kind of maintain those. But at the same time, there's an interesting correlation where people with high proportion of type 2 muscle fibers, they tend to be more susceptible to various diseases like diabetes and a number of different metabolic issues and early mortality. Now, that could be a case of maybe reverse causality because correlation is not causation, of course, but it's something that's sort of interesting at what's happening with that. And so anyway, so that's just something do we need to keep in mind as well. So that's kind of interesting with that. And then when it comes to these type IIx muscle fibers, they are pretty amazing. It's just that we don't have a lot of them. And get this, if you were to train, let's say you're going to be like, you're going to do weight training and everything, right? What do you think is going to happen with your type IIx muscle fibers? Are you going to get more of those or less of those? It turns out you get less of those. And in fact, when you stop training, your type 2a muscle fibers, if anything, you get a transition that goes more to the type IIx muscle fibers. And people have noticed this. In fact, I had noticed this myself. I used to train, I do like 100 miles a week, and I measured my vertical jump, and it wasn't super high. And then after I had a sabbatical of like running high miles, I wasn't doing, my strength was pretty much the same in terms of like my squats or deadlifts or whatever, but I did another vertical jump, and it went up like 4 inches just from not doing so many mileage. And it's probably because some of that proportion of the type IIx muscle fibers had to change. And so it's kind of an interesting thing that training actually decreased the proportion of type IIx muscle fibers to make it more of your type 2a. And then detraining kind of transitions things back over to that type IIx muscle fibers. So I would kind of wonder if maybe that's a reason why taking time off gives you maybe a higher PR, not because you're fully recovered, but because you're getting some of this transition that's going on. That's kind of speculation on my part, but maybe people already know that, but I'm just thinking about it. Now, we know about muscles pretty well already. I think you need to remember like the different phases of like dynamic muscle contraction, right? And so we have like our concentric phase, and that's of course your muscle shortening, right? So you have concentric, and it's really the angle of the joint, right? So if the joint angle is decreasing, right, then that's going to be the concentric. and is getting longer. That's the eccentric phase, right? So the lowering portion. And we think about on the sarcomere level what's happening, right? During the concentric movement, you're getting the shortening of those sarcomeres versus the eccentric. They're getting pulled apart, and then you have the importance of like a tighten to kind of prevent overstretching and things. And that eccentric, if it's too much weight or whatever, that causes more of the damage, like the Z-line streaming that we talked about. And that's, of course, if just the weight's really heavy and you can't pick it up, that's an eccentric motion. And there's actually some times where that type of training could be very beneficial for getting out of a plateau, for example. Okay, so now we talk a little bit about our force regulators, right? So we have things like the muscle spindles, and these are this intrafusal muscle fibers. There are gamma motor neurons as opposed to our alpha motor neurons. And they basically sense, they're the afferent motor neurons, and they're going to sense the muscle elongation and promotes the contraction of the antagonist muscle. And so that contraction is going to reduce like overstretch. And that's like with stretching, you have this proprioneuroceptive facilitation or PNF override. And it's interesting. And you've probably done this before. If you're stretching like your hamstrings or trying to touch your toes or something, and you get to that point of that stretch, and then if you do a contraction of your hamstrings, an isometric contraction really hard, and then you stretch again, you can go a little bit further, and that's kind of part of a force regulator with these muscle spindles. Another really interesting one is this Golgi tendon organ. I think this is fascinating because it has direct implication in training. And how you can basically lift more weight even when maybe you shouldn't, because this Golgi tendon organ it's located in the tendons, and it senses the tension, and it reduces the contractile strength via activation of an inhibitory response. Okay, so if you have like a ton of weight, let's say you're bench pressing, and there's like way more weight, and your spotter lets go of the bar. I don't know if you've experienced this before. Or even just like if you have like holding your arms out and someone just places a whole bunch of weight, your muscles, if there's all of a sudden a rapid amount of weight, your muscle literally just like you have this release of the contraction and activation of the antagonist muscles, and you have no strength. And that's because of this Golgi tendon organ, which is this reflex to protect your muscles from overstretching. Well, I guess from breaking, from causing all this damage. Now you could train over time to inhibit the Golgi tendon organ, and that's why you do things like with like really heavy weight, and you're learning how to do like an eccentric portion, and you're learning how to inhibit that Golgi tendon organ stimulus so that you can get closer to your max amount of weight. Now the danger, of course, is now you are at a higher possibility of maybe tearing a tendon or your muscle or something. But with training and things, it's another neat way that you can gain more strength because you're preventing that activation of antagonist muscles. So there's a number of factors that influence our contractile force, right? So you have, of course, your cross-sectional area, right? Like how big the muscle actually is. And then you also have your sarcomere length as well. That's going to be important because you have more of those cross-bridge formations, so you can pull those Z discs together. Another interesting one is the angle of pennation. So you have like your muscle fibers, and what is their angle? So if it's directly longitudinal, then you can put a lot of force on that, but you can only put so many muscles in series, basically, or not in series, but I guess on top of each other, not in this longitudinal way, and you can get a lot of force that way. But if you put your muscles at an angle, right, so it's kind of like at a pennation angle, right, then you can put more of those muscles in that pennation angle, and now you have a larger cross-sectional area, and now you're going to have a greater force contraction. So that's the benefit of having pennated muscles. And then, of course, another factor is your motor unit recruitment, right? When you activate your motor unit, well, how much of how strong of a stimulus was that motor unit? So you get how much calcium was released, right? Like and how strong are all those muscle fibers really contracting? That's going to be another part with that. So when it comes to just to maybe elaborate a little bit more on actually that pennation angle a little bit, this angle you have with you have your longitudinal axis, and it's along the entire muscle fibers. And of course, some of your muscle fibers really don't have any angles. But again, as I mentioned, there is an advantage, and maybe I can draw this out, or maybe just think about it. Like you have like your biceps; those are like fusiform or parallel. Your biceps brachii, and then you have like unipennate, which like I don't know, your extensor digitorum longus. But other ones like have a bipennate, like your rectus femoris, for example, is bipennate. But your deltoids are multipennate, and you can get more strength out of that whole cross-sectional area. And as you're training and you increase cross-sectional area, then you can get stronger. So that's going to give you more strength with that. Now, a big one when it is, especially when you first start training, is just your neural adaptations that are going to occur with resistance training. So when you very first start, in general, the increases in strength that you very first start seeing, most of those are neural adaptations, and that largely occurs before you start getting hypertrophy. And that's the first one. And it's possible that you're going to get a little bit of like various muscle adaptations and things that happen, like remodeling of the architecture of the muscle fibers. But often people notice they get quite a bit stronger really quickly when they very first start training, and that only can be explained by the neural adaptations. Now, advanced lifters who've been training for a long time, it's possible they also get some other more sophisticated neural adaptations, and they're able to like when they're already at the ceiling in terms of like the amount of muscle mass they can actually put on, they can still start getting stronger and stronger, even though the muscle is not getting bigger and bigger. And actually, it's very interesting that the relationship, the correlation between muscle size and strength is not as strong as you would expect. You would expect it to just be totally linear, right? That the bigger the muscle, the stronger the individual. But that's really not what's happening. That tends to only happen in a certain part of the training cycle. Like with the beginners, there's no correlation at all because most of the strength comes from the neural adaptations. And then your advanced lifters, people already are maxing out on how big they're getting, but yet they can continue getting stronger and stronger. And that's probably because of these neural adaptations, right? And you can look at some of these with like the different types of specific activity that's happening when you're looking at like EMG activity. There's this thing called the SAID principle, which is the specific adaptation induced demand principle. So things like plyometrics and other activities are going to really increase your strength. And also with more of the neural adaptations, you're getting more of the selective motor unit recruitment, and that it doesn't really, I guess in some ways it kind of violates the size principle, but it's a selective motor unit recruitment. And that happens with training. And another big one that increases your force strength is your post-activation potentiation. And this one is very fascinating. And people have noticed, you've noticed this, if you go to the gym and if you do your warm-up sets, then and then you lift like close to your one repetition max, and then you go back down and you see how many times you can do, say 80% of your RM, you often can do a number of more repetitions after you did all of that. After you warmed up, you lifted something really heavy, and then you came back. And that's partly because of this phenomenon called post-activation potentiation, where you get calcium that gets into the cytosol. It gets kind of on the titin molecule and a protein and a number of other areas, and make it so your sarcomeres can basically contract greater. There's a number of interesting mechanisms that's going on. Another part of the neural adaptations is also decreasing the activation of antagonistic muscles. So if you're trying to lift something, you don't want, like if you're curling something, you want your biceps to be activated, not your triceps, which would be the antagonistic muscle. You want to decrease that. And then you also get an increased reflex of the motor unit contraction. So if you have some of the spindle stretch reflex, for example, you can try this also when you're jumping or if you're doing squats. If you have your squats here, if you just go down nice and slow and then back up, yeah, it's really hard. You can feel that. But if you notice that if you go down, you kind of just drop that last little bit, boom, you kind of get that spring up. That's because you're activating that spindle reflex. And it lasts for a long enough period of time that you can even do this when you're going in for like a max deadlift or something, and you come over here and you just fall down, and then you grab that bar and then you can pick it up. There's different training you can do this, even with bench press. You can try to get activate that spindle reflex, and it's there for just a little bit. So that's another little trick you can do once now you understand muscle physiology a little bit more. Then you also have your synchronization of the motor units, because remember, you have multiple motor units, and typically these are, you don't want to get fatigue, and so they're switching back and forth to different motor units and everything. But if you can get synchronization perfectly, you're going to be able to get a lot stronger. You lift more weight, even though your muscle fibers or something didn't necessarily get bigger, but you have better synchronization, right? And then, of course, as I mentioned, let's see, the neural disinhibition of the Golgi tendon organ, that's a big one. And then of course, there's just the movement of the specificity, right? Like if you know that movement, say bench press, a squat, clean, all those things, being really proficient in that movement and practicing that over and over again is going to really increase the amount of weight that you can lift, even though you may not have gotten technically stronger, but you just have better form. It's that specificity pattern overall. So these are some of the neat things about how you can improve the strength and muscle performance and all of your muscle fast twitch fibers. Now, when it comes to creating this force, remember that it all originates in the sarcomere, right? That's the origination of the longitudinal force, and then that force has to be transmitted laterally, force transmission, as we show here, right, through to your endomysium and then the paramysium, and then your entire epimysium, and then to the tendon, and then onto the bone. And when we think about just that contractile element of, like, say, the sarcomere, we need to remember there's a lot of architecture that are within the muscle fibers, and all of those are basically acted like in series and put together in ways to allow this force production and force transmission, both active force and passive force, that allows the muscles to work in unison and in this really synergistic, amazing way. So it's really amazing when you consider the architecture of the muscle fibers and how we get the force going and then transfer all that force out laterally. So hope that was understandable, and thank you for your time.